Right, so now we're going to finish up hearing as a sense, and we'll do vestibular next by talking about the neural pathways. So now that we have transmuted a mechanical signal, the sound wave, into a neural signal with an action potential from the spiral ganglion cells traveling through the vestibulocochlear nerve, where does it go from there? And what I want to point out, because I've been doing a lot of contrasting and comparing with the visual system, is that unlike the visual system, where those retinal ganglion cells sent out their axons that went through the optic nerve, optic chiasm, optic tract, and they went directly to the thalamus. The first synapse was in the lateral geniculate nucleus of the thalamus, not so with sound. So it's different here. So from the vestibulocochlear nerve, cranial nerve 8, coming from the cochlea, carrying those action potentials from the spiral ganglion cells. Our first stop is actually going to be in the medulla oblongata. So it's called this cochlear nuclear complex just because it's a bunch of neurons that are having to do with receiving signals from the cochlea. The next thing is interesting. So watch how some of these signals here are going to go to each side. This is new and different. So after the medulla, everything is binaural, meaning everything as we move up through the different regions of the brain, we are getting input from both ears on each side. So this is very different from visual information. Now, it wasn't one eye to one side and the other eye to the other side. It was your visual field, right? So the left visual field went to the right side of the brain and the right visual field went to the left. So no one part of your brain actually has the full visual picture, but it's different for hearing. So once we get out of the medulla, there is some crossing or decussation. So that is kind of a vocabulary word that you might have picked up on in the textbook. They cross or decussate, and so we have both ipsilateral and contralateral signals coming up as we move through the brain. So continuing moving up then from the medulla, we're moving up into the pons, and we're going to find a place called the superior olive. Somebody thought it looked like an olive. So the superior olive. So again, I'm reminding you this receives inputs from both sides, and the job of the superior olive in the pons is to figure out where the sound is coming from. We call that localization. So when you hear a sound, you're getting information in the olive from both ears. So it's going to look at two things. It's going to look at in which ear is it louder? Because if it's louder on this side than it is on this side, well, then the sound is probably coming from over in this direction. It doesn't rely on that a ton, however, because sometimes, as I've recently discovered, you have decreased hearing in one ear compared to the other. So it mostly uses timing. So sound waves travel quite quickly, but not as fast as the speed of light, as you know, and our brains are actually able to perceive the slight differences. So if something's coming from over here, it's going to reach this ear before it reaches this ear. The sound waves will. And so your brain is able to look at that and calculate and then decide, is it over here? Is it kind of here? Is it halfway in between? Is it over there? So it's pretty amazing, actually, that we're able to localize sound as well as we can. As we'll see in lab this week, we're really good at horizontal localization because we have one ear on each side. We do not have an ear up here and an ear up there. So when it comes to the ability to tell whether something is coming from above or below, that's a little bit more difficult, but you can also understand why we didn't really evolve to have those senses because the ground is below us, and usually there aren't a ton of things flying around up above us that we need to worry about. So it's mostly that horizontal plane that a potential predator might be coming from. So that's the superior olive. It's going to look at the differences in timing and a little bit difference in volume to figure out where things are. From the pons, then, we're going to continue our upward trajectory. Next stop is the inferior colliculus of the midbrain tectum. So now we're up in the midbrain, and you might remember when we learned about different regions of the brain that the midbrain is responsible for a lot of visual and auditory reflexes. Ta-da! So this is going to help us focus on sounds in the environment. So when these impulses travel to the inferior colliculus in the midbrain, that is responsible for things like turning, toward the direction of a loud noise, for example, or your eyes at least going in the direction of a loud noise. As you can imagine, that would be a good survival reflex to have. So midbrain auditory reflexes. From the midbrain, we finally get to the thalamus, right? So we had medulla, pons, superior olive, localization. Oh, look, I even put it down here, so we'll animate it. So from the vestibulocochlear nerve, we went up to the cochlear nucleus in the medulla. Some of the tracts, axons crossed or decussated. So then by the time we get to the superior olive in the pons, we are binaural. We're getting signals from both ears, so we're able to localize. And then the signals go up to the midbrain for our auditory reflexes, and then finally we get up to the thalamus, and this time it's the medial geniculate nucleus. So for vision it was the lateral geniculate nucleus, and for hearing it is the medial, which is here and here. Right? So most of our senses have a stopover in the thalamus, which you remember is kind of the triage center for sensory information, figuring out whether to pay attention to it or suppress it, and then where in the brain is it going to go. So from the medial geniculate nucleus, that's a mouthful, of the thalamus, we're going to send impulses, we're going to send axons out to the primary auditory cortex, or A1. So same construct is vision, right? So from the thalamus, we went to the primary visual cortex, or V1. In this case, for hearing from the thalamus, we go to A1, the primary auditory cortex, which is located in the temporal lobe, which is very convenient. It's very close to the ears. And again, this is going to receive raw data of just what the actual sounds are, what are the frequencies, what are the amplitudes, and then we're going to have to use association areas to figure out what those sounds actually are and to interpret them. So there are lots of auditory association areas. The textbook doesn't go into a lot of detail. There does appear to be a dorsal stream, which is focused with where, and a ventral stream, which is focused with what. But as you read what they have in the textbook, there's a lot of kind of overlap and things seem kind of similar. So this is a very complex thing to make sense of what we are hearing and figuring out what it is and what it means. Now there is one particular area that we're going to come back to later in the course, which is called Wernicke's area, and that is very important for understanding speech and language that comes into us through our hearing and, interestingly, through some of our other senses as well. So just like for vision, raw data came into the primary visual area, and then we had association areas. Same thing happens for hearing, which is our auditory sense, right? Auditory, yeah. Now, just like vision, those association areas can be tricked. So sound effects for movies are a great example of this. So this image shows somebody taking a shirt and going boom, boom, boom with it to make that sound of a beating heart. So there's an optional media assignment where you can watch this little YouTube video about how they do a variety of different sound effects for videos. So we can trick the auditory association areas, especially if the information we're getting from our other senses, especially our visual senses, right? If our eyes are telling us it's a heart beating, then we're much more likely to be like, oh yeah, that's a heart beating, right? That's what I'm hearing. So kind of cool stuff there. In summary, from the hair cells of the cochlea, whose stereocilia bend, opening mechanically gated ion channels, potassium rushes in, depolarizing the cell, which causes voltage-gated calcium channels to open. Calcium binds to the snare complex, causes the exocytosis of glutamate into the synapse with the spiral ganglion cells. And then they send action potentials down their axons, which become the vestibulocochlear nerve. That first travels to the medulla, where then we're going to have some decussations or some crossing, and then it goes up to the pons, where we have the superior olive that's going to take that information to figure out where the sound is coming from. Then it goes up to the inferior colliculus in the midbrain for those auditory reflexes. Then up to the medial geniculate nucleus of the thalamus, which is then going to send the impulses onto the primary auditory cortex in the temporal lobe. Then from there, we're going to involve auditory association areas so we can make sense and interpret the information that's coming in. Remember, Wernicke's area is going to come back to say hello to us later in the unit for speech and language. And just like the visual association areas, we can trick the auditory association areas with sound effects. All right, that's it for hearing. Up next is vestibular sense.